Okay, if it's not one thing, it's another. So I just did this recording and realized that only a minute of it actually recorded. So I got to keep an eye on that. So hopefully it all works this time. All right, so what I want to go over today, or in this video, is looking at this lac operon worksheet. And this is a whole bunch of mutants that were used by Jacob and Minot back in the 40s, I think. It might have even been as late as the 60s, to figure out how genes are regulated. And so this is the first example of where they figured out how genes are regulated, or that genes were regulated at all. And a lot of what they figured out is still informed how we think about how genes are regulated now. So just to remind you, this is the lac operon, and the lac operon has transcription start site, promoter, operator, has this I gene over here that makes the repressor. It has lac Z, lac Y, and lac A, so Z, Y, A. Lac Z makes beta-galactosidase, Y makes permease, A makes transacetylase, but we're not going to worry about A. What we also know about the lac operon is that in the absence of lactose, these genes are not expressed. That's because the I gene makes a repressor. That repressor binds the operator and it turns the gene off. So the operon, or gene, is off. In the presence of lactose, the repressor still makes, the I gene still makes the repressor, still have a promoter, still have an operator, still have a transcription start site. You have these genes, but now we have plus lactose. So we got a bunch of lactose. Lactose binds onto the repressor. It can't bind the operator. So then RNA polymerase can bind, transcribe the gene, and make all three of those proteins. So first it makes the RNA and then it makes the proteins. All right, so that's a quick review of the lac operon. Now we're going to go back and look at some of these mutants, and this is how they figured out that the lac operon had all these different elements. So the I, the P, the O, the Z, the Y, the A, they figured that out by making mutants and seeing what happens. Now let me also go back to just one more review point, and that is that the lac operon is inducible. So if we measure gene expression starts out low, then we add lactose, and gene expression increases tremendously. So gene expression is induced, or this is an inducible operon. So it's off in the absence of lactose and on in the presence. And that phenotype is called inducible. So that's why they looked at this, because we go from off to on. And on means the gene's expressed, that it's transcribed and translated and all that nonsense. So it's a good system for figuring out how genes get turned on. It's a very simple switch. You just add some lactose and the thing goes on. So that's why we study it, because it's a very simple on-off switch. All right, so the initial phenotype of the operon, so the wild-type phenotype of how this operon works, is that in the absence of lactose, so the inducer is absent, the genes are off, and we're just looking at Z and Y, so this is the Z gene and the Y gene. Give me a Y, come on. Okay, so that's a Y. It's off in the absence of inducer, on in the presence of the inducer. Then they look at a couple of different mutants. So if you mutate each of the structural genes by itself, so you just make that Z minus. So just think of any kind of mutation so that you can't measure the protein product. So in all these things, they're measuring the protein product. If you make a mutation in the middle of the Z gene so that it makes it so that the protein isn't made, then you can't see that protein and it'll always be off. So a Z minus mutant is always off. Alright, give me a pen here so I can write, please. Alright, I guess we'll do it in red. Well, we should be able to do blue. Alright. Sometimes now the pen doesn't want to work. So this one's Z's always off. But Y plus is normal and everything else is normal, so it's going to be off and then on in the absence and presence of lactose. Letter C, Y is mutated, Z is normal, so and everything else is normal, so it's off in the absence and on in the presence of inducer. I minus, so now we don't have a repressor because I stands for the repressor. So if we have a gene and we have minus lactose, plus lactose, now we're I minus. So these are two different situations. So this, no repressor. If there's no repressor, then there's nothing to turn it off, so it's on. In the presence of lactose, it's on even if there is a repressor, but we don't have a repressor, so it's always on. So that's the phenotype of I minus. Everything's always on. The IS mutation in E, that's a super repressor. So that's a repressor that it makes a repressor, but this repressor is insensitive to lactose. So it actually has a mutation in the spot where lactose binds. So even in the absence of lactose, the repressor binds. But even in the presence of lactose, if we put lactose in there, lactose would like to bind onto that repressor and cause it to let go. But it can't let go because the repressor has a mutation in the spot that usually binds lactose. So it's a super repressor, so it represses all the time. And I like to think of it as the repressor binds and it gets stuck. Because once it binds onto the operator, so this is the operator. Oh, that should be the operator. Let's put the quoter down here. And that's the operator, so the repressor binds the operator. And it gets stuck. So if it gets stuck, and it always binds the operator, then the gene is always off. Now here's another mutant in F. That's a mutation in the operator. So instead of being O plus being wild type, it's OC. That's a mutation where the repressor cannot bind onto the operator. So it's the operator that's mutated. So the repressor binds the operator because the operator has a specific DNA sequence. If you change that DNA sequence by a mutation, then the repressor can't bind it anymore. So whether you have a regular repressor or a super repressor, doesn't matter, it can't bind. If the repressor can't bind to the operator, then the genes are always on. G, what happens if you have a super repressor and an OC mutant? Now let me just so let's talk about OC here real quick. So now we've got an OC mutant. Promoter's fine. Everything else is fine. Even your repressor might be fine. So now this I gene makes a normal repressor, but there's some kind of mutation in that operator so the repressor cannot bind. So then the gene's always on. But what if we make this a super repressor? So now we got a super repressor. This super repressor has a mutation in it so that it can't respond to lactose. And so in theory, this repressor should bind the operator and turn the gene off all the time. But this is the part of the protein that binds the DNA, and it can't bind the constitutive operator. So the constitutive operator kind of overrules the super repressor. So anything where it's always on, the word for that is constitutive. Versus inducible. So inducible means off and then on, and constitutive means always on. And OC is a constitutive mutant, so even in the presence of the super repressor, it's always on. The last, so H is one where this is just showing that there's nothing new here, but what this is showing is we've got a constitutive operator, and Z is normal, so Z is always on, but Y is mutant. So even though it might always be transcribed, there's a mutation in the Y gene so that you can't measure whether or not it's on. So it always appears off. So it's just a Y minus mutant. If you have a Z minus mutant or a Y minus mutant, mutations in the structural genes themselves, they will always appear to be off because they're mutated. I minus D for now, so this is letter I, just think of it as I minus. And so it's going to have the same phenotype as I minus up here. And so that phenotype is that things are always on. That's also a constitutive mutant, so because it's because the gene's always on, and constitutive always means on. As we'll see later, the D part of I minus D is that the D is dominant, and so we'll see in an example. We'll go down to letter P. We'll see that the I minus D mutation is dominant over I plus. All right, so I'm going to check every now and then to make sure I'm still recording. Alright, so the next one, now we start, all the rest of them are these partial diploids. So now we're going to have a situation where we have a bacterial cell. The bacterial cell has its normal chromosome, but then we can put another piece of DNA in there as part of a plasmid, or sometimes it's just a fragment of DNA. And you could, in this fragment of DNA, could have its own genes. And let's do the first example. So the first example is letter J. We're going to have I minus for one of them. So we'll make this I minus. And that one also has, everything else is normal. Alright, so that's that one. And then the other gene, and it doesn't matter which one you put on top. That one was P plus O plus minus minus. So plus plus minus minus. So now when we put two of these copies of this operon in a cell, we can measure where the gene products come from. Is it coming from the chromosomal gene, or is it coming from the plasmid gene, or is it coming from the blue gene, or is it coming from the red gene? So, what would happen in this case? Well, Z plus and Y plus, the only functional copies of those genes are coming from the red DNA. So this, if we're going to make anything, it's got to come from here. But this has an I minus repressor. The I minus phenotype is that those genes are always on. So should those genes always be on? Well, it turns out that this guy makes its normal repressor, and this repressor can act on that operator. It can also act on its own operator. But the fact that it can act on both, that it can go across and act on a different piece of DNA, we say that that acts in trans. And so in all these examples, really the repressor is the only thing we're going to talk about acting in trans. But we have to think about what is the evidence that the repressor acts in trans. And it's this kind of experiment that shows that if we supply the repressor on one piece of DNA and show that it acts on the operator on another piece of DNA, that means it acts in trans. And what would that phenotype look like? It would mean that the top gene so the Z plus and the Y plus that are on the top, if they were controlled only by the I minus that it's connected to, then they'd always be on. But if this I plus can act across party lines there and go up onto the top line, then Z plus and Y plus will be regulated by that repressor. And so they'll be inducible. And so that's what they see when they do the experiment. And so that says that I plus can act in trans. If it couldn't act in trans, if I only acted in cis, then the I plus on the bottom strand could not, strand's not the right word, on the bottom piece of DNA could not regulate the top DNA. But it can, so it acts in trans. K is another example of something acting in trans. The thing that's different here is now we're separating out where the Z plus comes from and where the Y plus comes from. So that's pretty useful. So now we know if we get Z gene product, we know it came from the top, and if we get Y gene product, we know it came from the bottom. All right. So and everything else is normal. So now, except for there's an I minus on the top. So if we have this I plus, if it can act in trans, it should affect both Z and Y, and it does. If I plus acted in cis, then only Y, because Y is connected to it, only Y would be regulated in that way, and Z would always be on. But it doesn't act in cis, it acts in trans. All right. In L, we've got a super repressor on top, a normal repressor on bottom, Z plus on top, Y plus on bottom. And the question again is, so now the super repressor turns things off all the time. If it acted only in cis, it would bind onto the operator on top, and it would turn Z plus off all the time. But the bottom gene, so Y plus coming from the bottom, if it was controlled by I plus, it would be inducible. So if the I gene, if the repressor acted in cis, then Z would be controlled by IS, and Y would be controlled by I plus, and they'd have different pattern. But what happens is the IS binds to the operator on both sides and turns both of them off all the time. That's because the super repressor, that's actually dominant to wild type as well, and it turns the operator off. So even if there's normal repressor that binds sometimes and lets go in the presence of lactose, once the super repressor binds, it gets stuck and it turns the gene off, and it's always off, as long as you have a normal operator. Letter M, we got a constitutive mutant on top, and it's connected to y plus. Got a normal operator on the bottom, and it's connected to z plus. The constitutive, if the operator could act in trans, then maybe the OC mutant would affect both y and z. Or maybe the O plus would affect both y and z. But what happens with the operator is that the OC operator controls y, so y is always on, because up here we saw that the phenotype of OC was always on. But the z plus, which is connected to the O plus operator, that is inducible in a normal way. So the O plus operator, just like in wild type, the phenotype of that is inducible, off and then on. So what we see in M is that OC controls y and O plus controls z, and they don't cross over. O plus does not cross over, crossing over is not the best word, but O plus does not act in trans. It does not come up here and affect y plus, and OC does not affect z plus. Because they act in cis. So what acting in cis means is we've got OC here. This might be a different example. We've got O plus here. And the reason we have Z minus and Y minus is so we can tell if we make Z, Z's got to come from the top. If we make Y, Y's got to come from the bottom. We know that the phenotype of OC is that a gene is always on, but it only affects Z plus. So this is always on. O plus affects this one, so it's inducible because it acts like a normal operator. So Y is inducible and Z is constitutive because it acts in cis. This doesn't control this and this doesn't control that. It doesn't work. So the idea is to look at these things and understand which example tells you what acts in cis and what acts in trans. N is another example. So now we've got OC connected to Z plus, O plus connected to Y plus. But now we've got this I minus. But we know that I plus makes a repressor, makes a bunch of copies of it. That repressor can act in trans. So as long as you get the I plus from somewhere, then you've got I plus. It doesn't matter where it comes from. But OC is going to keep Z on all the time, because now Z is connected to OC, and Y plus is connected to the normal operator, so it's going to be inducible. In O, the super repressor can bind onto the operator that's on top that's controlling Z, so Z is always off. But the super repressor can't bind onto the constitutive operator, because the constitutive operator can't bind onto anything. And so Y, because Y is connected to the constitutive operator, it will be on all the time. This is an interesting one, P. So now we've got the I minus D mutant, and we're putting it together with I plus. And remember we said the I minus D phenotype was that it was constitutive. So things that are connected to I minus D, or even if they're not connected, where's I minus D? So here, up in I, the phenotype for I minus D by itself is the same as I minus, meaning it doesn't make a functional repressor, and so the genes are always on. Alright, so what else do we have? The top is P plus O, everything's normal except for Y. So this is P plus, O plus, Z plus, Y minus. The bottom, everything is normal except for Z. So there's Z minus, Y plus, O plus, P plus. And what's the phenotype here? So Z is up top, and it's connected to I minus D. So if I minus D acts in cis, it would only affect Z plus, and Z plus would be on all the time. And if I plus acted in cis, then it would affect Y plus, and Y plus would be inducible. Well, it turns out that what happens here is that both Z plus and Y plus are on all the time. And that's because this I minus D makes a mutant protein. And the mutant protein makes a complex with the wild type protein. So this is wild type and this is the minus D mutant. And this is non-functional. So this is evidence that the protein acts as a multi-subunit complex, and that and what happens is this I minus D acts as what we call a poison subunit, and it poisons that complex. And so that's what the I minus D is. So in the I minus D, so it's an I minus mutant where everything should be on all the time. But if it was just I minus, and you didn't make any repressor at all from the I gene, then the I plus would make a normal repressor and it would bind both operators and act normally. But if you have I minus D, which is dominant over I plus, it does this. It makes a non-functional repressor, and that repressor can't bind to either operator because it's messed up. So, and it acts in trans, so everything's on all the time. The next couple are kind of messed up. So Q and R are messed up a little bit because this has two operators and this has two Z's. So let's not worry about Q or R. But now let's look at S. So S has a promoter mutation. So it makes a normal repressor, so that's fine. Both of the operators are normal, so that's fine. But there's a P minus mutation on the top. Well, if you don't have a promoter, you can't transcribe the gene no matter what. So, and promoters act in cis. Because that promoter is connected to both Z and Y, then both of them are off all the time. Because you don't have a promoter, so you can't, RNA polymerase can't bind. In T, the last one, this is showing that the promoter acts in cis. So Y is connected to a mutant promoter, so Y is always off. But Z is connected to a normal promoter, so it's normal. And in this case, it also has a normal operator, so it's going to be inducible. So the promoter and the operator always act in cis, and the repressor acts in trans. So just to belabor that point, what we're showing is that there is a transcription start site, then there are DNA sequences that are connected to that site. These sequences act in cis, so they're called cis-acting sequences. And then we have a repressor that can come from anywhere. So we could have this repressor coming off some gene over here, makes a repressor. That repressor can bind the operator, and so it's a trans-acting factor. Now, when we look at eukaryotic gene regulation, this is where we start. We start with the idea that there are cis-acting sequences and trans-acting factors, and we look and say, well, is it the same as bacteria, or is it different, or what's going on? Okay, so hopefully that all recorded. That's the infamous sheet here. Don't worry about those. There are some examples of this in chapter 16 in the problems, and so take a look at those. I might have taken those out because those were hard to read, and so maybe I should put those back in. But anyway, so if you can do this sheet, then you got it made. All right, so we'll stop it there.